Well, good morning, dear students. Today we will be tell about metabolism of proteins. No, and lecture we dedicate exchange of proteins in the body. We will be tell about immortal possibility of our components, broken of proteins in our body, place of it breakdown, proteolysis in gastrointestinal system, importance of it, and total pathways of amino acid metabolism in our cells. No, and our body is not infinitive, hasn't infinitive lifetime, not immortal, because not immortal all of its components. We are open systems and all our components constantly change. It takes place destroying and metabolism and resynthesis of our components, and after any time, for example, after any months, all our components will be fully exchanged. No, and all of it organized in metabolic map, where you can orientate now relatively. No, and now, before this moment, you studied metabolism of carbohydrates, metabolism of lipids. It interconnected from carbohydrates can synthesize lipids, and partially it used for possibility synthesis of any components. And all metabolic pathways finished by citric cycle, TCA cycle, with donor of hydrogens for electron transport chain and synthesis of water, and associated metabolism of ATP. I hope you know it very good. But in this picture will be added metabolism of proteins. Proteins constantly exchange, broken to amino acids, and in cells, amino acids used for synthesis of own proteins, for it necessary information of DNA, RNA, and amino acids can go to the providing of energy. Carbon skeleton of amino acid go to the citric cycle across any intermediate total substances, and ammonia will be exit from our body as urea. Well, about it some later. And all of it you can see combine with citric cycle as central metabolic centrum for synthesis of ATP and providing of energy. And also amino acid skeleton can used for synthesis of lipids, and any multiple of amino acids, carbon skeleton, can go to the synthesis of glucose and glycogenic amino acid. Now it pathways proteins broken to amino acids and go to the citric cycle, or can go to the synthesis of glucose, pathway for synthesis of nucleotides and lipids. And ammonia as additional factor exit as urea. And proteins in our body in constant dynamic state. And every day you can see any persons of proteins broken, degradate, and must be resynthesized. This structure of primary, secondary, tertiary, and quaternary structure, well known for you, it not constant structure. And after 1 month, almost not single protein will be present, which present now. And after some months, our body will be practically fully, no, by any exception. Hemoglobin have lifetime 4 months. Elastin much more. But mainly, usually, lifetime of proteins, some days, some minutes sometimes. And it was determined, discovered in 40th years, 40th years of last century by using of labeled nitrogen. It Rittenberg, Urey, and Horkheimer, you can see, provide evidence that proteins exchange it. For example, using of labeled nitrogen led to appearance of expression only any part, but other part will be accumulated in the body as proteins. And why proteins degrade? No, because proteins are chemical active substances, and in chemical active environment take place constant spontaneous or maybe planned modification, and it will lead to destroying of its function, missing of its function, spontaneous denaturation, or maybe synthesizing incorrectly. And cells constantly present in situation when necessary to select. Either will be divide, proliferate, or maybe constantly work in this state, or maybe must be activate apoptosis. And it constantly dynamic state. And lifetime of proteins you can see in different tissues. Lifetime maybe, of course, it's summary. Any proteins more long time, any less, but intermediate you can see in liver not more than 1 day, in kidney some more, but usually it's not too long. And where proteins destroyed? It may be extracorporeal, usually it's gastrointestinal tract. When I ask students where take place protein digestion, in gastrointestinal tract, maybe in the body, no, it's very often a problem question. But intracorporeal proteolysis can be intracellular, and mainly in our body take place intracellular proteolytic proteolysis, and it can be lysosomal mainly and proteasomal. Near half percent of all proteins broken by lysosomes, and near half of all proteins broken by proteasomes. And extracellular also take place, but it may be sometimes special cases. For example, blood coagulation, it may be complement immune defense, or maybe constantly exchange of extracellular matrix. But extracellular matrix, collagen and elastin, it long time living proteins, and it exchange may be problem, but mainly in intracellular. No, and extracorporeal, of course, important for compensation of broken and compensation of amino acids, which will be constantly exchanged. And about lysosomal proteolysis. Lysosomal proteolysis is discovered by Christian de Dieu, and it was determined presence of any parts of cells, vascular structures, with a high concentration of hydrolytic enzymes, with acid pH. One membrane surrounded structures. No, and the multiple proteins destroyed, including big structures. For example, mitochondria also broken by lysosomal proteolysis. And lysosomes is part of Golgi complex, separated from it, and consists a lot of proteins which will be active in acid pH. And acid pH is the result of working of ATP pumps, which will be pumped protons instead of sodium and potassium ions, and additionally chloride instead of carbonic acid. No, and proteolytic enzymes, it consists some tens hydrolytic enzymes, not only proteolytic, but it nucleases, glycosidases, lipases, other, and all of it optimal pH, acid pH. It's possible to tell about stomach of cells. Different mechanism of action. It's cysteine proteolytic enzymes, serine, aspartate, multiple complex activity, provide effective breakdown of all components. It not every time successfully, for any bacteria can be alive, but usually it full digestion of all components. No, and lysosomes. In lysosomes usually take place breakdown of substances which will be appeared by phagocytosis and endocytosis, which will be appeared by vesicles surrounded by cellular membranes. Or maybe intracellular structures, including mitochondria, for example. Now you can see it receptors specifically recognize any particles, any substances, associate created imperfect endosome, but after absorption with lysosomes will be appeared active complex with full digestion. In ideal case, of course, full digestion before amino acids, and for proteins before amino acids, for other polymers before monomers. But its mechanism, by the way, lysosomal proteolysis, effective mechanism for demonstration of antigens. For example, bacteria, antibacterial immunity depend on lysosomal proteolysis, absorption of bacteria broken, and specific antigens, specific sequence of amino acids demonstrated and associated with major histocompatibility complex, second type, will be demonstrated at the surface of antigens presentated cells. It will be recognized by immune sensitive lymphocytes. It's about participation of lysosomes, not only full digestion of components, but participation in immune demonstration, presentation of antigens. Proteosomal proteolysis. It was described at the end of 19th century by Keiji Tanaka, which will be discovered ATP-dependent proteolytic processes. For example, lysosomal proteolysis, it not used ATP for breakdown, but proteosomal proteolysis used ATP for breakdown, and for it used specific organelles, proteasomes, with size 26 S. You can remind acid Svedberg units, velocity of sedimentation in ultracentrifuge. It structure, symmetric structure with very intensive activity of proteolytic activity in the intermediate channel created by subunits, proteolytic, and recognizing and modified leads and proteins which will be have specific label. Its specific label, it's low molecular weight protein which called ubiquitin, and ubiquitinated protein with high activity will be broken by catalytic subunits of proteasome. Possible two pathways. Both variants will be possible, and each of parts of its proteasomal structure consists a lot of its complex of proteins. No, not only enzymes, it recognizing, defolding, and unfolding, and other parts which will be preparing a polypeptide chain for breakdown by intermediate proteolytic part. And about ubiquitin, a very important factor for proteolytic, for proteasomal digestion, it label of proteins which called ubiquitin. Very interesting protein, consists 76 amino acids. And if you listen about idea about molecular clocks, it changing of amino acids by mutations per any period of time, and usually at near 200 years of life take place changing of one amino acid per 200 amino acid. And if you will be compared the same proteins, for example, hemoglobin, hemoglobin consists in different vertebrates. For example, humans, humans, and frogs also everywhere present. But comparison of primary structure lead to possibility to compare evolutionary trees. And it takes place constant mutation. But for ubiquitin, molecular clock practically stop. For example, in yeast, one single cellular organisms, and humans, 10 power 14 number of cells. Differences, primary structure practically the same. Difference only 3 amino acids. Mutation in this protein lead to impossibility of life. You can see. And ubiquitination, interaction of carbon C term with substrate protein. And for it necessary specific enzyme system. 3 types of enzymes. E1 enzyme first, ubiquitin activated enzyme by ATP. Active complex used by second, ubiquitin conjugated enzyme, which will be facilitate association of ubiquitin with proteins which might be broken, and ubiquitin ligase. And we have some hundreds different ubiquitin ligases specific for different groups of proteins, which will be ubiquitinate, create polyubiquitin chain And these labeled proteins will be very quickly broken by proteasomes. Also possible non-proteolytic pathway for proteasomes if will be modify different groups, different other groups of lysine. It's some specific multi-potential protein which will be regulate metabolism, not only proteolysis, but also labeling of proteins for secretion and other very important protein, a very interesting protein which will be now intensively investigated. Also in cells in associated membrane, premembrane, undermembrane space work specific calcium-dependent proteins, proteolytic enzymes, which call calpains, diol proteases, and active in neutral pH. And now with regulation of proteolysis, you can see multiple functions. It breakdown of proteins, it regulation of cell cycle control, reparation of DNA, participation in development, the other antigen presentation, and destroying of its proteolytic pathways lead to some pathological events, consequences. For example, it can be lead to cancer, can be lead to dysfunction of inflammation, and serious neurological disorders, for example, Alzheimer and Parkinson and other diseases. And now you can see summary of proteolytic events in cells. It lysosomal proteolysis, it proteasomal proteolysis, and multiple functions, multiple events, and all of it important for a constant effective function of cells. Destroying of it all of it part lead to some problems with cells. No, and you can see proteolysis in modulation of cell metabolism. For example, function of any enzyme depend on its synthesis, which very often depend on signals which will be appeared from membrane, and it possible reversible reversible activation, deactivation, usually phosphorylation, for example, can be interaction by regulatory enzymes, can be allosteric regulation, can be for example, any enzyme work with corresponding substrate, and it also influences on it, and also breakdown of it enzyme. It's very effective for regulation of its activity. And totally for intracellular events in cells, very important, heat breakdown and amino acids and proteins constantly broken, and result amino acids constantly exchange used for synthesis. But partially it can used for any specific synthesis, for example, synthesis of any hormones, for example, epinephrine, and any other, serotonin, for example, can used for appearance of energy, input in TCA cycle, but for it necessary missing of amino group, and carbon skeleton used for broken to carbon dioxide and water. No, hydrogen go to the water and carbon dioxide the result of decarboxylation. And amino acids can go to the synthesis of glucose, gluconeogenesis. And it pool of amino acids, it very dynamical exchange pool. Practically free amino acids, not storage in cell. It constantly dynamically exchange. and necessary to get amino acids from diet. Without it, constant using of amino acids for any non-protein synthesis and constantly modification of its proteins lead to decreasing of pool of proteins, and for it necessary its compensation. And destroying of breakdown lead to accumulation of proteins in cells. Inhibition of normal proteolysis lead to so-called poisoning by proteins, and it lead to some serious dysfunction of the body. It's so-called prion diseases and Alzheimer's disease, amyloidosis, and oncogenic problems. Why? Because cells constantly take place in dynamic state when appeared any proliferative signals. It constantly appeared, and if will be it signals will be regulation of protein synthesis, oncogenic proteins. If it will be accumulate, it will be lead to non-planet proliferation. If will be ineffective breakdown of it. It's decreasing of proteolytic activity, but increasing of proteolytic activity also will be non-pleasure, because system of proteolysis and system of proliferation of cells depend on activation signals, oncogenic proteins, and function of anti-oncogenic proteins, suppressor proteins, tumor suppressing proteins. And very active breakdown of suppressor proteins also will be lead to non-planet proliferation. Both variants will be lead to increasing of oncogenic transformation, cancer. It balance between it two processes very important. Now you can see a result of accumulation of protein poisoning, accumulation of amyloid fibrils, structures, fibrillar proteins created by non-digested proteins, which will be accumulated in connective tissue, and now it prion proteins, which will be lead to prion diseases. It proteins in central nervous system, which work provide metabolism in neurons. Maybe it function unknown fully, provide interaction between membrane and cytoskeleton, or maybe transfer signals from membranes. And differences in energy of folding between alpha helix and beta sheets not too high. And the normal configuration for prion disease, prion protein, it alpha helix. But if it will be folded in beta sheets, it will be more stable, more stable for proteolysis, more hydrophobic protein, which will be additionally transformate the normal of protein with normal alpha helix to beta sheets. In cells will be accumulate non-digested proteins, prion proteins, and at last it will be lead to dysfunction of neurons, activation of apoptosis, and accumulation of digested broken cells, so-called spongiose encephalopathia. Now we can see normal monomeric soluble sensitive proteolytic enzymes and mainly alpha helical. Not scrapy, so-called first time of discovered. Multimeric, multiple insoluble protease resistant and mainly beta sheet. Now you can see Now you can see lifetime of usual lifetime of proteins depend on amino acid sequence. And amino terminal residue, if it will be consist any amino acids, it will lead to stability of its protein. You can see any amino acids very quickly lead to destroying. You can see proteolysis depend on primary structure of proteins and depend on activity of proteolytic system. Now again return to its question and necessity of compensation of amino acids from diet. And again, in summary, amino acid proteins constantly used for resynthesis of proteins, but later carbon skeleton after deamination, ammonia go to the synthesis of urea, and carbon skeleton go to the citric cycle. Between two H cycles, citric cycle and urea cycle, both of it, by the way, Krebs cycles, take place interconnection across any intermediate substances, total for both of it. And for compensation of used amino acids necessary, we must to get proteins in diet. And if it compensation will be absent, it will be lead to constant decreasing of nitrogen-containing substance in the body. And comparison between entrance of nitrogen-containing substances, proteins, and exit from the body on nitrogen-containing proteins, nitrogen-containing substance, called nitrogen balance. Differences between intake and exit of nitrogen-containing substances. And absolutely minimum, it was provided by in 40th years of 20th years of last century in volunteers, and it was determined that if organism cannot to get proteins in diet, per day will be broken any level of proteins, and it's absolutely minimum near 40 grams per day in comfort condition, without intensive physical exercises, and in comfort condition, not cold, not and without stresses. But if input will be more than output, it's a positive nitrogen balance. When it will take place? In growing infants or babies, pregnancy, and recovering after serious disease, traumas. If input will be less than output, or maybe diet, absence or inefficiency of proteins in diet, or maybe any serious physical exercises or stress, any other senescence, it will be every time exit more than input, and in this case it will be negative nitrogen balance. No, and in optimal conditions, nitrogen equilibrium, when input and output the same. For adults, it's up to 1 gram per day. For babies, it may be more, necessary more than 1 gram, and for newborn, it may be 3 grams per kilogram of body weight. By the way, up to 1 gram in adults, 1 gram per kilogram per day. For example, usually it for optimal conditions, it near 80 to 100 grams per day. But important factor for protein nutritions, it presence of essential amino acids. Proteins can be so-called completeness proteins or perfectness proteins for diet. It depends on essential amino acids. Essential amino acids, amino acids which we cannot synthesize. And a list of absolutely essential amino acids you can to see. It amino acids with relatively complex radicals. It hydrophobic amino acids, isoleucine, leucine, valine, methionine. It positively charged amino acid lysine. No, and it aromatic amino acids, phenylalanine, tryptophan, and threonine, also relatively complex, OH with methyl group. And any amino acid can synthesize in our body. But it can synthesize from absolutely essential amino acids. For example, cysteine can synthesize from methionine, and tyrosine from phenylalanine. It conditionally essential amino acids. When enough methionine and tyrosine and phenylalanine, it will be non-essential amino acid. But if amino acid will be inefficient, it will be essential. And amino acids which can synthesize in our body, but not concentration is less than necessary. It's all used for any other processes. For example, arginine. Arginine can synthesize, work in urea cycle, but better if you will be getting additionally from diet, partially essential. And histidine for adults, it's not every time essential, but for babies it's essential. No, and non-essential, which can synthesize in our body, alanine, asparagine, aspartate, glutamate, glutamine, glycine, relatively simple amino acids. But usually what amino acids more important for human body, essential or non-essential? Usual answer, it's of course essential. But reality is next. For synthesis of non-essential amino acids, necessary to have special enzyme systems. Enzymes, it's necessary to have correspondent genes, correspondent a lot of energy used for its synthesis. And evolution tried to avoid of not such essential processes for life. And if in evolution presence enzymes and any energy-dependent processes for its synthesis, it tell about importance of amino acids. If we can to get amino acids from diet, from environment, not necessary to have special enzyme systems for its synthesis. Of course, non-essential amino acids, evolutionary more important. But it's a paradoxal point of view. Well, about appearance of amino acids from diet. We can to get proteins and amino acids from gastrointestinal tract. For it we have special stomach, intestine, duodenum, intestine, small intestine, you can to see different parts of gastrointestinal tract. And we must to get amino acids. If proteins, foreign proteins will be appeared in by a void of gastrointestinal tract, directly appeared in the body, it will be lead to effective immune answer against it proteins. And amino acids hasn't immunological properties, answers, and the breakdown of proteins take place beginning from stomach. And we have different types of cells. In stomach we have acid pH. It result of function of this function of parietal cells, which will be secreted HCl, and the chief cells, which will be secreted pepsinogens. All of its synthesis depend on function of specific so-called enteroendocrine cells, which have different name and distributed in different parts of gastrointestinal tract. For example, in the stomach take place G cells and the D cells, which will be provide regulation of synthesis of HCl and pepsin. And pepsin synthesizes pepsinogen when pepsinogen is inactive enzyme, and activation provided by HCl and outer activation. All one of main factors for synthesis of HCl and pepsinogen, it innervation by vagus, and which will be secreted acetylcholine, and acetylcholine activate antral G cells and so-called enterochromaffinic-like cells. G cells synthesize gastrin. Gastrin is a very effective activator of parietal cells, which will be synthesized HCl, and also it activate cells which will be synthesize histamine, enterochromaffinic like cells. By gastrin actively influence on it, totally it lead to effective synthesis of HCl, and also it lead to specific cells in duodenum and antral place, which will be lead to synthesis of secretin, which will be activate of chief cells with synthesis of pepsinogen. And also after appearance of acidification of stomach content, signal from acidification lead to regulative cells in duodenum, which will be synthesized also specific regulative polypeptides, which will be, for example, secretin activate synthesis of bicarbonate by pancreas, and will be synthesized cholecystokinin, which will be lead to a contraction of gallbladder for increasing secretion of exit of bile, and motilin, which will be activate motoric of stomach and gastrointestinal. Not totally you can to see it, for example, sensitivity to content of digested proteins. It G cell, which will be synthesized gastrin. Gastrin activate D cell, which will be activate enterochromaffinic-like cells, which will be synthesize histamine. Histamine and gastrin activate synthesis of HCl by parietal cells. But D cells, when acidity will be increased, will be synthesized so-called gastrin inhibitor will be lead to activation of D cells, which will be activate somatostatin. It's inhibitor of electrochromophenic cell-like cells. I hope on and all of the picture will be developed in antral part of stomach. Now it's about synthesis, mechanism of synthesis of HCl. It depends on interaction of carbon dioxide and water activated by carbon hydrase. Result will be carbonic acid, which will be dissociated on proton and ion of bicarbonate. Protons will be pumped in luminogastric content by corresponding pump at antiimport of protons and potassium ion. It's ATP used process, and acidification will lead to activation of counter-charged ions. But counterflow of bicarbonate ions, and instead of it will be adsorption of chlorine ions, and lead to compensation of positive charge by HCl. But constantly HCl practically absent its structure. It content of ions of chlorine and HCl. And all of it will be activated by acetylcholine vagal regulation, histamine by enterochromophenic cells, and gastrin by G cells. And gastric acidity, now you can see a table of different variants. Total acidity is up to 60 millimole per liter. Less than 40, from 40 to 60. Less than 40 hypoacidosis, more than 60 is hyperacidosis. And acidity depends on HCl plus any other non-HCl acid component, but mainly HCl. And pH depends on free HCl. But free HCl can be sharply decreased after feeding. Now, for example, we can see before milling, concentration of protons relatively high, but after feeding, it HCl sharply decrease. Buffer activity by foods very increase. By gradually it will lead to activation of synthesis of protons, and pH will be gradually increased, but exit of foods and proteolysis of proteins will be lead to gradually decrease buffer activity of foods and returning pH to acid. Now you can see it millimoles. It volumes of in inner space, and pH of not pH but equivalence of acidity, and synthesis of HCl will gradually decrease. And after exit of stomach and stomach content to duodenum, it will be lead to activation of synthesis of bicarbonates by pancreas, and synthesis of which will be activated by secretin, and partially cholecystokinin will be activated process, and cholecystokinin will be activate contraction of gallbladder. And now we can see about specificity of proteins, of enzymes which will be digest of proteins. And for example, in stomach, synthesis synthesize pepsinogen. Pepsin gene will be transformed to pepsin, active pepsin, by digestion of peptide 42 amino acids and opening of active site. And pepsin can to digest mainly hydrophobic and aromatic amino acids. In duodenum will be work trypsin and chymotrypsin and any other proteolytic enzymes. Also synthesize and inactive precursors, trypsinogen, chymotrypsinogen, propeptidases, other, and it can to digest different amino acid sequences. Trypsin can to digest peptide bonds created by lysine and arginine. Chymotrypsin digest also aromatic and hydrophobic amino acids, phenylalanine, tryptophan, tyrosine. You can to see has similarity with pepsin, but mechanism action will be different. For example, phenylalanine will be point of action for chymotrypsin and pepsin, but phenylalanine has two peptide bonds, one from amino end and other from carboxy end. Carboxyl group created and amino group created. And pepsin can to digest peptide bond created by amino group of phenylalanine. And result will be phenylalanine on amino end of digested peptide. And for example, now glycine on carboxy end of other peptide. But chymotrypsin will be digest phenylalanine from carboxy end. And phenylalanine will be on carboxy end of new peptide, and on amino end will be now alanine. And the same mechanism action for trypsin, chymotrypsin and trypsin, it's serine proteolytic enzymes, serine in the active site, and will be digest on carboxy end of target amino acid. It will be lysine on carboxy end. Now you can to see about specificity of different proteolytic enzymes. And peptide bond is not some less strong covalent bond than other covalent bonds. It partially double distance between carbon and nitrogen, some less than single peptide bonds, and it's more sensitive to nucleophilic attack. But without proteolytic enzymes, lifetime of peptide bond, you can to see it very long, and nucleophilic attack by proteolytic enzymes provide very effective breakdown by seconds and some less milliseconds with digestion of peptide bonds. No, usually different variants nucleophilic substitution by active parts of proteolytic enzymes. We talked about mechanism action of enzymes. It every time must be electron donor, electron acceptor, nucleophilic or electrophilic attack on substrate. Now we can to see one of variants of it. And about activation of proteolytic enzymes. We told before, proteolytic enzymes synthesize as inactive precursors. Pepsinogen, inactive precursor of pepsin, trypsinogen, inactive precursor of trypsin, chymotrypsinogen, inactive precursor of chymotrypsin, and other. And the total activator, for example, in duodenum, it enteropeptidase or enterokinase enzyme activated by cells in duodenum, and it enzyme which will be activate of transformate inactive trypsinogen to trypsin. by digestion of short peptide, 6 amino acids, and it will be lead to forming of active site of trypsin, and trypsin by its activity will be transformed chymotrypsinogen to chymotrypsin by proteolytic activity, inactive proelastase to elastase, for example, procarboxypeptidase to carboxypeptidase, prolipase to lipase by limited proteolysis, not full digestion of protein, but it limited digestion by selective, most sensitive peptide bonds. So for example, now you can see transformations of inactive trypsinogen to trypsin by enteropeptidase. 6 amino acid will be digested from amino end of it, and transformation to active pepsin, and later active trypsin, sorry. And the trypsin, active trypsin, will be transformed inactive chymotrypsinogen to active chymotrypsin across intermediate steps. At first, digestion of one peptide bond and the transformation of chymotrypsinogen to p-chymotrypsin. It's active but unstable. And next, digestion of short two amino acids, digestion one and second. Next, maturation of p-chymotrypsin, transformation to alpha-chymotrypsin, which active and stable, relatively stable. And what is next about transformations of proteins? Amino acids, at first proteins digested to amino acids in lumen and oligopeptides, and oligopeptides digested by enzyme proteolytic activity associated with intestinal wall cells, and possible digestion before D and 3 peptides. It also can be adsorbed, and it complex adsorbed into adenum and go to the blood. Now, in summary, stomach digestion, acid hydrolysis, activation in stomach and small intestine, and digestion before maximum D and 3 peptides, but mainly amino acids. And amino acids, adsorption of amino acids, very similar with adsorption of glucose. For it necessary specific co-transporters with sodium ions, and the main factor for adsorption is sodium-potassium pump, which will be used ATP for secretion of sodium from cells, and instead of it, adsorption of potassium ions. And it leads to inefficiency of sodium and across sodium channels and co-associated absorption of amino acids. Amino acids appeared in cells, and later transporters of basolateral membranes will be go to the blood. Now you can do the summary. It total structure. Digestion by pepsin on a relatively big part of peptides, oligo and polypeptides, chymotrypsin, elastase, carboxypeptidase A and B, trypsin, digest to amino acids, and amino acids by brush border amino acid transporters will be adsorbed in cells, and later BLM, basolateral amino acid transporters will be transported amino acids to blood. Mechanism of function of intestinal mucous layer, by the way, very similar with function of mucous layer of other mucous containing organs. Intestinal adsorption, it ATP dependent. Lungs absorption, the same or isoenzyme systems provide absorption of useful substances from blood to blood. Absorption exit from any substances. For example, anion transporters, cation transporters, multi-drugs and toxin transporters. And for example, secretion of any substances to bile, also it system with similar functions, and very often it isoenzyme or isoprotein, which will be have similarities in function. No, and about intracellular transformations of proteins. We told that proteins digested to amino acids and go to the cells. And by the way, entrance of amino acids to cells depend on function of glutathione and some so-called gamma glutamyl transpeptidase. It's part of glutathione. Glutathione is very interesting three peptide which consist cysteine, glycine, and glutamate. And the glutamate interact with glycine by carboxy group of radical, and glutamyl transpeptidase take place digestion of glutamate and interact with amino acids which must be absorbed. Cysteine glycine, part of glutathione, will be digested, broken to glycine and cysteine, and can be reabsorbed by ATP-dependent process. And complex glutamate and amino acids adsorbed to cells. Glutamate transformed to oxyproline, cyclic structure, and later by ATP-dependent transformed to glutamate and re-synthesize glutathione, ready to next absorption of next amino acid. And amino acid appeared in cell. For each cycle necessary to use again to see 3 ATP. And next, total pathways of amino acid. Usually it go to the protein synthesis, about it I will be tell some later, but for entrance to TCA cycle, necessary to avoid of amino group. And now please short break, now near 5-7 minutes, and after it we will be continue. Так. Well, and now about intracellular transformations of amino acids. Deamination. For missing of amino group, in nature present four main types of deamination. It oxygen-free, it reductive, hydrolytic, and indirect appeared in time when oxygen was absent in atmosphere, and oxidative deamination. You can to see reductive, it addition of hydrogen result deamination ammonia and saturated acid, or really fatty acid, fatty-like acid, reduced, full reduced. Hydrolytic, missing of ammonia and addition of water in water environment, appearance of alpha hydroxy acid. Indirect, for it necessary two carbons together, missing of ammonia and appearance of non-saturated bond, non-saturated fatty, non-saturated acid. And oxidative, addition of oxygen, result again ammonia plus alpha keto acid. For humans, realize mainly, absolutely, practically, it oxidative, but a little partially indirect and hydrolytic. No, indirect a little, but mainly oxidative. And oxidative deamination, possible two variants. It one of it using of enzymes oxidases. It flavin dependent enzymes, flavin associated enzymes, and oxidases can transfer of hydrogen directly to molecular oxygen. Result will be hydrogen peroxide. But after oxidation of missing of two hydrogens from amino acid, will be appearance of amino acid, alpha amino acid. And amino acid is a very unstable structure, and in water environment will be forming of alpha keto group and free ammonia. Oxidizers, you can to see indirectly provide a structure which ready to hydrolyzate. But activity of it enzymes very low, very low. Practically for big part of amino acids we have oxidases, but its activity it's non-compatible with intensity of its amino acid deamination. But one amino acid deaminate much more effectively, much more intensive. It's glutamate. Glutamate deaminate by dehydrogenase. In contrast with FAD oxidases, it NAD and it dehydrogenases. And the mechanism practically is the same. Missing of two hydrogens, transfer of NAD, or sometimes with NAD phosphate, result amino acid, which will be actively, spontaneously hydrolyzate with appearance of very good known, I hope, alpha ketoglutarate. Alpha ketoglutarate is intermediate substance for TCA cycle, tetric cycle. And intensity of its process much more than flow of concentration of glutamate. And it led to idea that possible indirect transfer of ammonia when amino acids will be deaminate not directly by oxidases, but take place transfer of amino group on alpha ketoglutarate, and alpha ketoglutarate will be dominated by glutamate dehydrogenase. And it will be successfully discovered with opening of special group of enzymes which call aminotransferases. And aminotransferases provide domination of different amino acids, transfer on alpha-ketoglutarate as substrate of amino groups, and alpha-keta acid, which will be appeared from amino acids, will be used for different, for example, go to the TCA cycle. And glutamate will be intensively dominated later with appearance re-synthesis of alpha-ketoglutarate. It's called indirect domination. And it appears that first step of catabolism of most amino acids is transamination, when amino acids will be transformed to alpha-keta acids, alpha-ketoglutarate to glutamate, and acceptor may be not only alpha-ketoglutarate, but maybe also oxaloacetate. No, and amino acid transferases, or more shortly transaminases, you can to see it important factor for transfer of amino acid. And all of its enzymes, transaminases or aminotransferases, it complex enzymes which consist non-protein part. It pyridoxal phosphate. It derivative of vitamin pyridoxine. Pyridoxine must be it derivative of pyridine ring, consists pyridine ring and alcohol group on one carbon. And it must be oxidized to aldehyde group, pyridoxal phosphate. And pyridoxal phosphate will be interact with amino group, will be transformed to pyridoxamine phosphate and keta group, alpha-keta acid. And later pyridoxamine phosphate of non-protein part of aminotransferases will be re-transformed to pyridoxal phosphate and with transfer of amino group to any alpha-keta acid with appearance of new amino acid, very often it glutamate. And looking please, if we will be returned to domination of glutamate, you can to see alpha-ketoglutarate is result of domination, but equilibrium, constant of equilibrium near 1, and increasing of concentration of ammonia will be lead to displacement of equilibrium to glutamate. And but alpha-ketoglutarate, as you know, it intermediate substance of TCA cycle. And if will be increasing of ammonia, it will be lead to decreasing of alpha-ketoglutarate in cycle. And what function of TCA cycle? It donor of hydrogens to respiratory chain. And inhibition, increasing of ammonia will be lead to inhibition of intensity of TCA cycle and decreasing of ATP synthesis. And what is most sensitive organ for ATP? It brain, and for nervous system it's very important. TCA cycle is very important, and ammonia is very toxic substance for nervous tissue. It's about toxic effect of ammonia. Ammonia is inhibitor of TCA cycle. Now you can to see active site of aminotransferases. It derivative of pyridoxine, vitamin B6, which can be provide interaction between active sites of between amino acids and alpha-ketone acid. And activity of aminotransferases, it's important indicator of situation in different tissues. Aminotransferases, it's intracellular enzymes, and activity of aminotransferases in intercellular space and in blood is very low. But if will be any problems with nutrition and providing fuels and oxygen in any tissue, for example, infarction of different tissues, most important is for myocardial infarction. Activity of any of its enzymes will be increased. And if you remind about isoenzymes, set of isoenzymes in different organs can be different. And in heart muscle, concentration and activity of aspartate aminotransferase, which transportate amino group on oxaloacetate with the synthesis of aspartate, more than aminotransferase which transfer of amino acid to pyruvate. Next acceptor of amino groups can be pyruvate with synthesis of alanine. And if in blood will be increased activity of aspartate aminotransferase more than alanine aminotransferase, it indicator of problems with myocardial infarction, heart muscle. Myocardial infarction will be increasing, reasons of increasing. And if will be increase more alanine aminotransferase than aspartate aminotransferase, it usually indicator of liver dysfunction. For example, viral hepatitis. And transfer of ammonia, we told that ammonia is toxic because it's inhibitor of TCA cycle. And in tissues, including muscles and brain, metabolism of amino acids take place every time. But transport of ammonia between organs in free state is dangerous. By the way, using of ammonia and synthesis urea take place in liver. And from tissues, transport of ammonia must be in bound state. And in this case, we also call it glucose-alanine cycle, when first acceptor of ammonia in transamination will be pyruvate, alpha-ketone acid, and after transamination, pyruvate will be transformed to alanine. Amino acid will be transformed to alpha-ketone acids, which will be used for any other metabolic pathway. Alanine, after transamination from pyruvate, will be transported to liver and transformed to by transamination to pyruvate, returning to pyruvate, and accepted will be alpha-ketoglutarate. And alpha-ketoglutarate in liver will be very actively transformed after transamination, transformed to glutamate, and very actively deaminate with forming of ammonia, which will go to the urea. Pyruvate can be go to the glucose synthesis, resynthesis by gluconeogenesis. Glucose go to the tissues, and again transformate to pyruvate, which will be ready to bound with the next portion of ammonia. Each cycle, it called glucosalanine cycle. You can to see that each cycle very similar with the Cori cycle, transport of hydrogen from muscles in intensive working. But now it transport not hydrogen but ammonia. Now you can to see, but very similar with Cori cycle. Now you can to see it pyruvate as carrier glucosalanine cycle. And in liver, using of alpha ketoglutarate not such dangerous how it, for example, in brain or muscles. Alanine, which will be after result of transamination in muscles or brain, transfer in bound state to liver. In liver will be transamination returning to pyruvate, gluconeogenesis by blood to muscles or any other organs, and its cycle, and glutamate deaminate with forming of ammonia and go to the synthesis of urea. Glucose-alanine cycle. Ammonia very toxic, especially for brain, and in brain works special other system which will be actively adsorbed ammonia, and for it used glutamate. Alpha ketoglutarate acceptor of ammonia with forming of glutamate, but glutamate also can be acceptor of ammonia, next molecule of ammonia, by ATP, phosphoglutamate, and binding of ammonia with forming of glutamine. Glutamine consists two nitrogen atoms and glutamine transported by blood to liver, where it will be deaminate with forming resynthesis of glutamate and resynthesis of ammonia. Resynthesis of alpha ketoglutarate with multiple additional acceptor of ammonia. It's very effective mechanism of binding of ammonia. Partially it can go to the kidney, and deamidase, glutamine deamidase take place in kidney, and partially ammonia directly can exit to urine. Now you can to see glutamate plus after activation by using of ATP, glutamyl phosphate, binding of ammonia, glutamine, and in liver or in kidney, glutaminase will be transformed to glutamate again. All of it lead to absence of free ammonia, which can lead to toxic effect by inhibition of TCA cycle. Now, excretion of ammonia in human body, it excreted as urea, but environment influence on possible variants of excretion. For example, in terrestrial living organisms, usually excreted by urea, it ureatelic, so-called ureatelic organisms. But urea is very osmotic active substance, and it not for excretion of urea necessary a lot of water. And for organisms who can live to have period of development by limited exchange by substances, for example, it eggs period. It births, it no, and insects also. It excreted by bed-soluble uric acid. It uricatelic. But other side, it for living organisms who live in water. Not necessary to synthesize any specific substance, can be excreted by ammonia ions. It for one cellular organisms and yeast, for example, no, and up to invertebrates and fish. Now you can to see terrestrial vertebrates excreted ammonia as urea, aquatic vertebrates ammonia ions, and reptiles and birds excreted as uric acid, bed soluble. About it, more detail we will be studying some later. Thank you very much. And now, goodbye.